Marietta,
Great.
are you ready?
I do. Jill, I hear an echo on your end, but hopefully you hear me okay now.
I hear you great. Thank you.
Okay, perfect. I see some head nodding. Okay. So no disclosures outside of being an employee of Pfizer. I will go ahead and get started. So good afternoon, everyone. My name is Mariana Bruno. I'm the U.S. Medical Affairs Lead for the Pfizer Cardiac Amyloidosis team. I'm here today to talk to you about how the totality of our data and the clinical effectiveness of tafamidis merits an A rating. At Pfizer, we're proud to have pioneered treatment with tafamidis in this space. The breadth of our clinical data, coupled with real-world experience, is unparalleled. To start, it's helpful to ground the audience in the unprecedented results from the phase 3 ATTRACT study, which significantly reduced combination of all-cause mortality and CV-related hospitalizations versus placebo over 30 months, with a p-value that was highly statistically significant. This trial led to the breakthrough therapy designation and FDA approval of tafamidis as the only treatment for hereditary and wild-type ATTR-CM to reduce CV mortality and CV-related hospitalization with a safety profile similar to placebo, as reflected in the label. With more than a decade of research and 6 years of real-world experience, we have generated the most robust and consistent clinical evidence to date across NIA stage 1 to 3, which we believe supports an A rating. ATTRACT and its long-term extension study demonstrated a statistically significant reduction in all-cause mortality and CV-related hospitalization out to 5 years. To put this in context, for every 8 people treated with tafamidis, 1 death is prevented, and treatment of 4 people prevents 1 CV hospitalization with 30 months. I hope we can all agree that this profound impact on patient outcomes is not commonly seen in CV outcome trials. During the ICER Early Insights webinar series, ICER acknowledged an A rating for tafamidis if evaluated in the original trial population, but expressed less certainty for its effectiveness in the contemporary population, resulting in a B+ rating in the absence of a clear definition. We strongly disagree with this assessment. There's no basis to suggest uncertainty in the benefit of tafamidis in a contemporary ATTR-CM patient. On the contrary, in a pre-specified analysis of ATTRACT in NIA class 1 and 2 subgroup, a less sick population, if you will, and a reflection of patients diagnosed today, there was a significant 43% reduction in mortality in the primary analysis and a 44% reduction with nearly 5 years of follow-up. As a reminder to our audience, NIA class 1 and 2 patients accounted for 70% of the original ATTRACT population, and those class 1 and 2 patients performed consistently better than the overall population. Furthermore, most recently, a post-hoc analysis from ATTRACT and its long-term extension study presented at ESC showed a separation of survival curves in the first few months of tafamidis treatment in patients with NAC stage 1 disease, emphasizing the importance of early diagnosis and treatment. These robust, consistent clinical data also supported our recent real-world survival analysis from TAUS, which looked at patients treated and untreated with tafamidis. A sensitivity analysis reflecting a contemporary cohort comprising healthier patients in early stages of disease showed a 42-month survival rate of 86.3% in tafamidis-treated patients. Importantly, no new safety signals were identified. With this totality, depth, and breadth of experience, there is no scientific basis to conclude that tafamidis would not have similar efficacy and safety in patients diagnosed today. Furthermore, based on data I've described, tafamidis has been recognized with Class 1 recommendation in NIA stages 1 to 3 in the highest evidence rating for the AHA, ACC, HFSA heart failure guidelines. In closing, tafamidis is the only FDA-approved disease-modifying therapy for both hereditary and wild-type ATTR-CM to reduce mortality and CV hospitalization. Robust clinical and real-world evidence consistently demonstrates the net health benefit of tafamidis across NIA stages 1 to 3, including a healthier contemporary patient, along with a safety profile comparable to placebo. Tafamidis has redefined the treatment of ATTR-CM, dramatically improving patient outcomes and leading to early diagnosis. We absolutely welcome advancements and innovation in this disease area, and as a leader in this space, we look forward to continuing to empower efforts with stakeholders to advance this mission and early diagnosis for patients with this serious and progressive disease. Thank you for your attention.
Thank you, Mariana. Does the Midwest CEPAC or does the ICER staff have any comments or questions for Mariana?
Jill, I'd like to respond just to the ICER evidence rating.
Please go ahead.
So first, I don't want to suggest that this is the most obvious call between an A and a B plus. As Dr. Bruno notes, during our early insights, I made it clear that there is no question that tafamidis would deserve an A rating in its original population. The only difference between a B plus and an A is greater uncertainty as to magnitude of benefit. It allows for the possibility that the magnitude of benefit is as great in a current population. It allows for the possibility that the magnitude of benefit is even greater in a current population with earlier disease. Those are all possible with this. The issue is we have a randomized trial of tafamidis in a population that looks nothing like the people who are currently being treated. And so we have indirectness in the way we think about this to the current population. More than that, we have what happened with acoramidis, where it didn't show a mortality benefit in the current population. And then we have vutrisiran, which did show a mortality benefit with a different mechanism of action. All of that taken together led us to conclude that we could not reasonably say that we had high certainty in an earlier population of this large benefit. That should not be taken to mean that we think that there isn't a large benefit or that it shouldn't be used in that earlier population. This is an evidence statement about certainty, not an attempt to suggest that the drug doesn't work, that it shouldn't be used, that some other drug should be picked first. All of this is just a discussion of our certainty in the evidence ratings here, and we wouldn't put it beyond that. And we really don't think that it's worth a lot of focus on this. I think that Dr. Bruno is correct in saying that tafamidis in guidelines has been over and over again said to be standard of care for this, and it has changed the entire way that ATTR-CM is managed. And that's obviously a huge deal. Personally, I wouldn't focus a lot on our B plus versus A evidence rating, but I do understand the concern and confusion it causes. And so I wanted to answer this. Thanks.
Thank you, David. Rachel.
Sure. I wanted to ask Dr. Bruno about potential differences in efficacy between varying doses of tafamidis, right? Given the potentially significant impacts on costs for patients and the system, does Pfizer have additional data about the potential differences between the 80 milligram and 20 milligram doses? And does Pfizer have plans to do additional clinical trials to determine whether there's such an efficacy difference and what it might be?
Yeah, so thank you for the question. I think it's important to note, I think a key differentiation about, as a clinician, rationing is not the best option for patients. And the dosing is flat priced. And there's also no difference in the sizing of the capsule. So I think I just wanted to make sure that that was clear to the audience. We stand behind our analysis that looked at the LTE, where there's a greater mortality benefit for the patients in the 80 milligram. That is, I think, what is appropriate for patients. In fact, the FDA felt the same. And if there was any question, we trust in our regulators that they would have, required another study or analysis based on the current indication.
Actually, can I ask a little more about that, Dr. Bruno? So one of the things that concerned us was the lack of data on any other outcomes besides mortality from the long-term extension of the 20 milligram versus 80 milligram dosing. Do those data exist, and are they going to be published?
But when you look at the original track trial in the subgroup analysis in figure 3, you will see from a survival benefit with the 20 milligram, it loses statistical significance in the survival analysis. So, with the longer exposure of patients in ATTRACT with the LTE, there was a significant survival benefit with the 80 versus 20, with a 30% relative risk of death reduced. So the survival benefit with the tafamidis 80 milligram compared to the 20 was also significant, but it's important to recognize the greater benefit with the 80 milligram when all covariates were adjusted. So including risk of death, NT-proBNP, 6-minute walk test. When all those variables were adjusted, the benefit was still observed with the 80 milligram dose.
Understood. Our concerns on interpreting those data were that we don't have the flow of the number of patients and the patient characteristics of people in the 20 milligram arm. The published data only tell us the flow of the placebo arm and the 80 milligram arm of how many people went into the long-term extension from the 20 milligram and what those patients were like versus the patients who went through in the 80 milligrams. So that makes the LTE data trickier for us to interpret than they need to be. And we don't have data on hospitalizations and other cardiovascular events, which might be supporting data that maybe it's not just a blip on the mortality. And that's why I'm wondering if more will be published about this.
Not at this time, but when you do look at the LTE, you will see the number of patients that continued on LTE for the 20 milligram dose. I could certainly take that back into discussion with the team if we would be producing another analysis, but at this time, we do not have plans to.
Thanks. We did ask for those data. So just so everyone's aware, we requested additional data around this because we thought this was an important issue.
I agree. I think that's a really important issue too, especially since the absolute numbers were lower with 20 at 30 months on several different outcomes. The long-term extension data that separates the 80 and 20, we do need a lot more data on that. A survival curve would be informative. Are there any other questions?
I guess, could you just weigh in, if there is a flat pricing for the 80 and 20, why the, I guess, curiosity or speculation? If there is a single formulation of an 80 milligram dose, which certainly patients who are, I think all of us agree with this disease, we're at a place where patients are living longer, that the single formulation 61 milligram, which delivers the 81 equivalent 80 milligram, is more effective. But why the probing for the 20 milligram if it's flat priced?
It isn't flat priced. You can get the 20 milligram pills, the 20 milligram capsules for 1/4 the price of the 61 milligram. Just to understand it, this isn't random. There are formularies that are using the 20 milligram because it's 1/4 the price. We kept hearing about this. Do you have some reason to think we've got that wrong? Because I know we have people on the panel who are experienced with formularies that are using the 20 milligram dose.
Right. I think you pointed out the formulary makers. So this is a broader sort of conversation as it relates to potentially prior auth criteria, but the recommended dose, we do stand behind the analysis and the recommendation of the FDA, because it would need to be the 4/20 in order to deliver the more effective dose as it relates particularly around mortality.
Right. No, we understand. We recognize that those are both the FDA approved doses. And the reason this comes up is because there are places that are using 20 milligrams. And we were trying to understand both how cost effective it would be if 20 milligrams were as good as 80 milligrams and what the data are that 80 milligrams is better than 20 milligrams. And so that was why our report talked about this to some degree, because we got comments from various people asking us to look into that question, and we didn't feel we could adequately answer it.
Okay, I understand. I think that might be unique to one particular payer or potential health system, but I can certainly take that back on my end.
we heard it from at least 2 health systems, just to know.
Okay, I'll make note of that. And as a reminder, just to the group, I appreciate being here from a medical point of view, and we do stand behind the efficacy and safety of our data, and always have patients top of mind. So thank you for your time.
Thank you very much for being with us today. We really appreciate you being here and your input. Thank you. So it is top of the hour, 3 minutes. Till the top of the hour, we're going to break for lunch until 12:50, and then we'll come back for the votes. See you in 50 minutes.
Thank you.